Alright guys, so I thought I would run through. I've just put together a lecture for the week 5, third lecture for PSY4050 Quantitative Research Methods, and I thought I'd just give it a run through, help me practice it, and also work out the timing to make sure I can fit it into an hour for when I do give this lecture face to face in a couple of weeks. Okay, so what we are got quite a lot to cover in this, so I'll probably have to talk fast. This week, what we're going to do in this lecture, this week in lab classes, I'm going to finish outlining some foundational statistical concepts. The first 2 weeks focus more on that. The first lecture focus on research design, and then last week, or 2 weeks ago, the second lecture was, and the lab classes were about centre, measures of descriptive statistics, centre and spread, and also some foundational concepts such as distributions. I'm going to finish those off by talking about resampling issues and the distribution of sample statistics, essentially. It's kind of useful for us to know. Then we're going to do we're going to introduce some statistical analyses to test research questions about differences between mean scores, so group mean scores. Specifically, we're going to do the t-test and ANOVA this week, and to do that I'll outline what the t-distribution is, how it differs from z-distribution, define what the t-test is and different ways that can be used to assess differences between two group means. You usually use it for two group means. And then we'll go on and talk about ANOVA, or analysis of variance. You can use ANOVA to compare two group means or more. Mostly people use it when they've got 3 or more group means. And then we'll also cover post hoc testing, which you would need to do if you have a significant ANOVA to see where your significant differences lie. So by the end of this week, after the lab classes in this session, you should be able to choose tests of differences between sample means on the basis of the design of the study and how many groups you've got. Interpret and evaluate the results of t-test and ANOVA and form and report those results, and choose between and interpret results of post hoc tests for ANOVA. Okay, so quick recap though, a quick review of null hypothesis significance testing. I covered it at the end of the week 3 lecture, but it is a combination of, hopefully remember, a combination of the idea of testing a null hypothesis, that there is no difference between groups in the population between the scores, against an alternative hypothesis that the scores of groups are different. Okay, we use null hypothesis significance testing uses the p-value as an index of the weight of evidence against the null hypothesis. So we assume the null hypothesis is true, there's no difference, and we use the p-value to evaluate the weight of evidence against whether it's true or not. So by convention, the kind of threshold for significance in this discipline is set at p is less than or equal to 0.05. So that's what we have to refer that to the alpha level of significance. And the smaller the probability of getting your results, the more likely there is to be some sort of difference, and the more likely it is that the null hypothesis is not true. Okay. So last lecture we also discussed z-scores and the standard normal distribution. We only discussed these in relation to calculating individual z-scores and assessing how far above or below the mean they might sit, and the proportion of scores that might sit between different values. Comparing the scores of one individual against the whole population is generally not that interesting, I don't think. And it's unlikely to be the basis of a research question or research project that you're conducting. So we're more likely going to be interested in comparing groups or sample means, something like that, than individual scores. So that's what we're going to focus on mostly for this lecture. But firstly, before we start looking at tests, inferential tests that allow us to compare group means, such as the t-test and ANOVA, there are a couple of other foundational concepts it's useful to know about. So a Typically, a sample is drawn from a population with the goal of measuring something. So, a sample statistic is calculated, or we might model something using an inferential statistical model. Since our estimate, the statistic, so for example, the mean, is based on a sample, it might be in error. So the actual real population mean might be different to our sample mean. If we draw another sample, we might get a different, we more likely will get a different mean from another sample. So we're therefore interested in how different it might be from sample to sample. So a key concern in statistics is kind of sampling variability. Okay, because we're using that to quantify uncertainty. If we had lots of data, we could draw additional samples and observe the distribution of sample statistics directly, and we could see how much they vary. Yeah. Typically, though, we calculate our statistics or estimates using all the data that we have available. So additional samples from the population are not readily available. Okay. Right, so let me read some of the content of this slide. Sample statistic, as you know, is a metric calculated from sample data. The sampling distribution of a statistic is the frequency distribution of a sample statistic over many samples or resamples. As I just said, it's often unrealistic. We often can't do that. We just have our one sample and we can work out the mean of that. Resampling would be a lot of effort and in many cases prohibitive. Okay, the central limit theorem is a useful theorem. It's the tendency of the sampling distribution to take on the normal shape as sample increases. So what that means, the central limit theorem says that means, if we take means drawn from multiple samples and we plot their distribution, the distribution will resemble a bell-shaped curve, even if the source population is not normally distributed. That happens if we have a sample size that's large enough and the data don't depart from normality too much. So the central limit theorem has an important role in traditional statistics because it underlies the machinery of hypothesis testing. It allows us to sort of look at how much uncertainty there is around our estimates. Okay, so the other thing there on this slide is the standard error, and that is the variability of a sample statistic over many samples. So the central limit theorem is saying if we take loads of samples, we resample lots, and we plot the means of those samples, if we've got a large enough sample, they'll be normally distributed. The standard error, if we then worked out of that distribution, we worked out the standard deviation, that is what was called the standard error. It's the variability of a sample statistic over many samples. Okay, so let's talk a little bit more about the central limit theorem. As I said, if we take lots of samples and plot their means, the distribution will be normal, like on this slide. So you can see what's happening here, though. Imagine someone has kind of drawn, for some research, they've done multiple samples and they've worked out the means of each of the X bar showed there of each of those samples. So the first sample's got a mean of 102, the second 99, the third 104, 108, and so on. They work out then what the mean is from all those samples, the mean of the sample means, and the standard deviation. And you will find that as the distribution will be normally shaped as sample size increases. And you can see here that there's on this slide you've got the formula. It's got sigma sub x bar is equal to sigma x divided by the square root of m. You can see here the formula for the standard deviation for this sampling distribution of sample means, and this is what we would call the standard error. Effectively, it's this, if we did a lot of resampling, plotted the means, plotted the distribution, and worked out how much variability there is, the standard deviation is the standard error. Say a little bit more about that. So the standard error can be estimated using the standard deviation of the sample and the sample size. So that's pretty useful. The standard error is, so it's a single metric for the variability in the sampling distribution of a statistic, and we're able to estimate it, get good estimate of it using the standard deviation of our sample. So if we've only got one sample and the square root of the sample size. So we have an idea here. This is useful because we have an idea here how closely our statistic estimates how statistics are estimating population parameters without having to do lots of resampling. Okay, we can estimate the variability with information about our sample without having to do those prohibitive tests. Those tests that we probably can't do of taking, go get another sample of 100 people, go get another sample of 100 people. Pretty difficult to do that. So if we know how sample statistics such as group means are distributed, that helps when we're conducting inferential statistical tests such as comparing differences between means across groups, sort of tests we're going to do today. So the ideas I've just covered in the last few slides all relate to what we could call resampling. Okay. And as I stated previously, it's often the case that the data we have is all we have to work with, and resampling will be unrealistic and impractical. Okay. Had computing power been widely available over a century ago when statistics was in its infancy and being developed, statisticians could have made use of computers to conduct simulations and do computationally intensive resampling. Yeah, and that would have helped us understand how population statistics and samples are distributed. However, as computers weren't available, this wasn't the case. As statistics developed, we had to rely on the development of mathematical functions that we could use to approximate these kind of sampling distributions. So how are certain statistics such as the mean distributed? Okay, and the use of those sorts of functions has influenced the development of statistics moving forward since that time, and these sorts of things have then become deeply embedded in the discipline and the formulas, the statistical formulas we use for conducting inferential tests. So the formula on this slide is the formula for the standard error, and you can see it's similar to the standard deviation formula that was on the previous slide. In this case, it's the SE, the standard error, is equal to S, which is the sample standard deviation, divided by the square root of n. Okay, so we're getting a good idea just from our one sample of what the variability would be in means if we did lots of resampling. Okay, so the t distribution and its associated t-tests, the t distribution is obviously a probability model, and t-tests, to test for group means, they're functions that have been of the sort I'm talking about, that were developed early in statistics, that have been relied on heavily, and we're going to discuss the t distribution and t-test now. So the t distribution is actually a family of distributions where the shape changes based on the degrees of freedom. You'll see what I mean in a second. It is normally shaped, but it's flatter and longer in the tails. It's used extensively in depicting distributions of sample statistics, such as how the mean's distributed. And it's kind of useful then. If we know how mean scores are distributed and we're comparing two means, we can see the extent of overlap in their distribution, and we can use those to figure out probability. That's pretty interesting, pretty useful. So the accuracy of the t distribution in depicting behavior of a sample statistic, though, requires the distribution of that statistic to be normally shaped. We know from the central limit theorem, it's quite helpful. We know from central limit theorem that that sample statistics follow a kind of normal shape, essentially, in their distributions. Okay, so on this slide you can see how the t-distribution becomes more normal shaped as the degrees of freedom increases. So the red line there is a normal distribution, standard normal distribution, I guess. And the green and blue lines show the shape of the t-distribution with different degrees of freedom. So the green line there shows what the t-distribution looks like when you've only got 2 degrees of freedom. The blue line when you've got more degrees of freedom. So as the degrees of freedom increase, the t-distribution becomes more and more normally shaped. So this is all well and good, but you might be wondering what this adds beyond what we know from the normal or Z distribution. Okay, so if you recall from the last session, the standard normal distribution or Z distribution, for that we had this Z formula we could use to work out Z scores for individual scores and see how many standard distributions they sat above or below the mean. So the Z score for single scores, you can see at the top there, where Z equals X, whatever the score is, minus mu, the population mean, divided by the population standard deviation. And we could use the sample. We could use the sample to figure this out essentially. So it was X minus X bar, the sample mean, divided by the sample standard deviation. But as I've said a couple of times now, we're often interested in groups of scores and rather than individual scores, and we can obtain Z scores for groups using the second formula on the slide. So the Z score for sample means formula is Z is equal to X bar, which is the sample mean, minus mu, which is the population mean, divided by sigma of X over the square root of N, so divided by the population standard deviation, that would be. But there's a problem here with this formula. That problem is, in order to use that particular Z-score formula for working out group means, the population mean and the standard deviation must be known. And we rarely know what mu and sigma are. We're rarely in a position where we know what the population mean is and the actual standard population standard deviation. So not knowing the population mean is not such a problem, as we're usually testing a sample mean against some sort of hypothesized population mean, and that's the hypothesized population mean for some tests. You'll see that in a second with the one sample t test. Is we're often assuming that the population mean is 0. Okay, but not knowing the population standard deviation is a bit more of a problem. Instead of using the population standard deviation, we could use the sample standard deviation. We can calculate that, we work out our mean, and then we can work out how much scores vary around the mean, figure out our standard deviation for the sample. And we could use that as an estimate of the population standard deviation. But in order to do that, we need to know how sample standard deviations and variances are distributed. And unfortunately, they are positively skewed. Okay, so they look like this. They've got a long tail towards the higher numbers. And what this means is that more than 50% of sample standard deviations will fall below the mean. You can see the median here marked in green on the slide. So 50% of scores will be to the left of that line and 50% of scores will be to the right of that green line. And as you can see, when you've got a positively skewed distribution, those kind of more extreme values at the high end pull the mean up. Okay, so that's higher. The mean is actually higher than the median. So if we put the sample standard deviation into a Z-score formula for means, we're likely to get an inaccurate estimate of what the mean should be, as 50%, as more than 50% of our participants, our observations, will score or fall below the mean. So So what I'm saying is, if we put S, the sample standard deviation, into the z-score formula for group means, we're dividing by too small a number, and we'll get a z-score that is too big. Okay, the bigger the z-score, as you know, the more likely it is to be significant. We saw the other week with the empirical rule that about the further your z-score from the mean, the more likely it is to be significant. And 95% of scores fall in between plus or minus 1.96 standard deviations off the mean on a standard normal distribution. So the bigger the z-score we've got, we've used the sample standard deviation in our formula. That's kind of given us it's given us an inaccurate estimate of what the mean should be, because more than 50% of scores are now below the mean, and what we've got is a bigger z-score that's more likely to be significant. So what we could be finding is significant results when the null hypothesis is true. Okay, so we're making a type 1 error. Okay, so we're effectively underestimating what the standard deviation should be if we use this sample rather than the population. Okay. Right, so Z versus T. I think I just said this, but I'll read the top bit. So if we look at the Z formula, which is on the right of this slide, which is the one for group means, if we use the standard deviation to estimate the population standard deviation, then Z is likely to be too large, and we're more likely to find a significant result than we should have. Okay, unfortunately, the bottom of the second formula there is the T formula. I'll come to that in a second. So this problem of using, what happens if we've used the sample standard deviation rather than the population standard deviation, was solved by someone called William Gossett, who wrote under the name of student because he worked for the Guinness Brewery and they wouldn't let him publish under his own name. This is a long time ago, probably I think 1908, something like that. So he showed that if you substitute sample standard deviation for the population standard deviation, this leads to a particular sampling distribution known as the T distribution. Okay, so this is quite helpful. So this guy's done the work for us. He's done the resam. He's basically done loads of sampling and then used the sample standard deviation rather than population deviation, and he's looked at how that changes the shape of the standard normal distribution. What you get instead of a z-score is a t-score, okay, or a t-statistic, and the t-test is simply the z formula, as you can see there, but using the sample standard deviation instead of the population standard deviation. So we can evaluate, because he's worked out the probability model, what the shape of the distribution looks like for T. We can calculate T and evaluate it against tables of probabilities for the T distribution. So like the Z distribution, someone's worked out the probabilities in the model for us, save us having to do it. That's pretty good. I'm not sure how to do that. I would glad smart people than me have figured that out. So effectively, if we make the standard normal distribution flatter and wider, so it's shaped like the T distribution, probabilities change. Okay, but we have tables that tell us how those probabilities change, which is good. Right. Let's talk about t-test then. So the one sample t-test. So when we conduct a t-test, so we're using the t-test, we're using it to compare means usually, or we're comparing two groups. The t-test is a bit of a tricky one that I don't like explaining, because the formula changes. It's kind of a more simple test because you're comparing two means effectively. but it's trickier one to explain than a lot of other more complex statistical concepts, because the formula changes depending on the design or scenario that you're using. So when we conduct a t-test, we calculate a t-value and then we compare it against probabilities for the t-distribution, which is the underlying probability model for the t-test. And the probability is based on the degrees of freedom for our sample. So we look it up in tables, effectively, like we do a Z, like we do with Z distribution tables. As I've said, the most common use of the t-test is to look to see if two group means are significantly different from each other. But the formula changes depending on the research question. So we've got a one sample t-test, which I'm going to talk about first. Then we've got a repeated measures t-test, and then we've got an independent groups t-test. Okay, so the most basic is the one sample t-test, where you have one group mean, and you're testing it against the hypothesized population mean. So this hypothesized value for mu, the population mean, is usually 0. Okay, so as you can see on this slide, the formula simplifies. So we've got t equals x bar, sample mean, minus mu, the population mean, divided by the degrees of freedom. Okay, that'll give us our test t statistic. But if we've got one sample and we're seeing how different it is from some hypothesized mean, which we don't know, so we're assuming it's 0, then mu is 0. So this simplifies, because you've basically got x bar minus 0, which is just x bar. Yeah. So it simplifies to x bar divided by s over the square root of it. So the sample mean divided by sample standard deviation gives you 2. I don't know how useful this is, or I mean, maybe there are times when you might use this particular test. I don't know how often anyone uses this. I guess H0 could be that if you've got some sort of difference scores in a group, the H0 could be the proposed difference is 0 or something like that. But then I guess that'd be very similar to repeat measures design, which we'll see in a second. All right. Well, yeah, that is exactly what it is. Let me move on. So one sample t-test, it's that formula essentially. We're looking at whether a group mean, the mean scores on a group that we've taken a sample from, differs from a hypothesized population mean that's usually 0. Limited utility, I think. Can't think of research designs that's helpful. Repeated measures t-test is essentially a one sample t-test but calculated on difference scores. Okay, so if you look at this kind of table here, we've got our participants under the column subject, so we've got 1, 2, 3, 4 going on, and then we've measured them on we've given them some sort of test or scale or measure, treatment, manipulation before time x1, and we've measured each participant's score, so 5, 7, 3, 9 for different participants, and then we've measured them at a later time after, and we've recorded what their score is on the second occasion, 7, 8, 5, 12. So this could be, I guess this could be, well, maybe we're testing some sort of drug. Maybe it's for anxiety. So we test their anxiety, look at what their anxiety scores are before, and then we a week or 2 later, maybe we look at what their anxiety scores after. Yeah. The fourth column there is labeled diff for difference, and that's basically the difference scores between the before and after treatments or conditions in this particular study. So we've got 5 and 7. If you've got 5 minus 7, that gives you a different score of -2. 7 and 8 gives you a difference. 7 minus 8 gives you a different score of -1. Difference here meaning you subtract one from the other. So you can see on the diff it says x1 minus x2, so that's what's been done there. Okay, and then for this difference column, for the different scores there, you can work out the mean difference, so the mean of x1 minus x2, and you can work out the standard deviation, the difference standard deviation. So what we want to know is if our sample mean difference, d bar, so our sample mean difference as opposed to population mean difference, where you want to know if it's come from a population of differences with a mean of mu d and a standard deviation of sigma d. So if we use the sample standard deviation to estimate the population standard deviation, then the t-test formula becomes the one shown below. And you can shown below that section, that bit of writing there. So you can see t equals d bar, which is the mean difference scores from our sample, minus mu of d, which is the hypothesized population difference, divided by the sample standard deviation. And if we don't know what the population difference is, then that's 0 again, like for the one sample t-test. So this formula simplifies to d bar, or the mean difference scores between the before and after condition, divided by the sample standard deviation. Okay, or the sample difference standard deviation. That's kind of what S sub d actually means there. All right, so that's the repeated measures t-test. So it's kind of essentially it's the same as a one sample t-test, but rather than a group mean, we're looking at different scores for participants between time 1 and time 2, so to speak. All right, independent group t-test. So here we have 2 independent samples coming from different populations. In theory, yeah. We want to know if there's a significant difference between our sample means, okay? And the null hypothesis here is there is no difference. So the population means have the same value. So 2 independent samples coming from populations with means mu1 and mu2. And H0, our null hypothesis, is if we take population mean 1, mu1, subtract population mean 2, mu2, then the result should be 0. They're no different, apart from a bit of sampling error. So we need to know about the sampling distribution and differences between means, and this is normally distributed. It turns out to be normally distributed. However, the formula changes with the independent groups t-test. Because the standard deviation is not equal to the difference between the population standard deviations, the distribution is more spread out, so we have to add the standard deviations together for the 2 groups that we're comparing. Okay, and when we do that, it's known as the variance sum law, essentially. So when we've got 2 groups for our t-test. When we add the standard deviation for group 1 and group 2, if you square that, it's the variance. It's known as the variance sum law. Just adding together the variances. So the formula for independent groups t-test looks like this on this slide. So t is equal to, in the numerator there, we've got in brackets mean of group 1, x bar 1 minus x bar 2. So that's the mean for sample mean for group 1 minus the sample mean for group 2. And then we've got minus, subtract from that, the population mean for group 1 minus the population mean for group 2. And those are in brackets as well. And then divided by, in the denominator, we've got the variance it's the standard deviation, it's the sum standard deviation for that group. So we've got a radical sign for square root, and under that we've got S1 squared divided by M1, which is the variance for group 1, plus S2 squared divided by M2, which is variance for group 2. Those have been added together, and we've taken the square root of them, which is basically just adding together two standard deviations. Note that I've put a little arrow there with usually equals 0. What that means is this: the second term in the numerator there, the mu1 minus mu2, that the population one is we as usual, we don't really know what they are, so that's often 0. But because we are testing against this hypothesized idea that mu1 minus 2 is 0, we can put that in there. Yeah. And if that's 0, again, this formula simplifies to the one shown at the bottom. So T is, you've got your sample mean 1 minus sample mean 2 divided by the summed standard deviations. Okay. Right. Slide 18. Okay, so I got a worked example here. Thankfully, it all fit on the slide without my image cutting you off here at the bottom. So, you're not going to have to remember these formulas or calculate these test statistics by hand. So SPSS will do all of this for you. Okay, but here is a worked example of an independent groups t-test on a very small made-up set of data, just to give you an idea of how it works. I think it's useful. It's useful to sort of just to develop your understanding of how this works, and to show you that although these formulas look really quite complicated, all we're doing is, you know, the concepts we covered 2 weeks ago around spread and things like that and center, we're using those in these formulas to infer things about differences between groups. All right. Okay. So for example, in this example, let's pretend we're looking at differences in scores on a maths test between two age groups. So 2 age groups have been given a maths test. A group of 6-year-olds, the same maths test being given to a group of 8-year-olds. We're looking to see whether there's a significant difference between these two group means in terms of the scores. And if you look there, so we've got I think we've got 9, have we? 10. Apologies. We have got 10 participants in each of our 2 groups, and we can work out the mean score for 6-year-old group, and it's 7. We can work out the standard deviation for that group, and the variance, which is obviously just square the standard deviation. We can do the same for 8-year-olds there as well, and we can see the mean is 7 for 6-year-olds, and the standard deviation is 1.83. The mean is 11 for 8-year-olds, and the standard deviation is 2.49. So the mean scores on this test are higher for 8-year-olds. Does that mean that's different, but is it significantly different? So we use our t-test to find out, and we plug those numbers into the formula. So we've got the mean of group 1, which is 7 minus 11 in the numerator, and then divided by what is the standard deviation. But if we plug in the number for variance, remember, if you square the standard deviation, you've got the variance. You can see where those numbers have come from there under the radical sign in the denominator. So we've got the variance figure for the 6-year-old group, 3.33 divided by 10. That's the number of individuals in that group, plus 6.22, the variance for the 8-year-old group, divided by 10, the number of people in that group. So if we do those calculations, what we get is a t-value of 4.09. If you look at those calculations, technically what we've got is a t-value of negative 4.09. Okay, routinely that's important, that the result comes out as a negative number. For the t-test, it doesn't matter though. The reason it's come out as negative is because we've taken it's a function of which group we took as group 1 and which is group 2. If we'd have put the 8-year-old group as group 1 and subtracted 6-year-olds from it, it would have come out. We've still got a value of 4.09. We would have got a positive 4.09 rather than negative 4.09. Okay, so with the t-test, don't routinely ignore negative signs, but for the t-test you can. Okay, in this case. So underneath that, you can see that that's how you would form a report. The result of the t-test. So you've got t, the test statistics, then in brackets you've got 18, which is the degrees of freedom, equals 4.09, and then you've got a comma, then you've got p for the p-value, equals 0.001. Okay, so about a 1 in 1,000 chance of getting a t-value that large or larger. So a difference in scores that large or larger. Okay. So that's a statistically significant result. Okay, the 18 for the degrees of freedom, where that comes from, if you remember, it comes from independent pieces of information. So in the first group for 6-year-olds, we've got 10 individuals. So when we work out the mean, we've got 9 independent pieces of information. Okay, so for the 6-year-old group, we've got 9 degrees of, we've got 9 degrees of freedom. For the 8-year-old group, same sort of thing. We've got 10 individuals. When we work out the mean, the only independent piece of information are the first 9. So we've got 2 groups of 9. Add them together gives you degrees of freedom of 18. So n-1 for the first group, n-2 for the second group. Add them together in this case. Okay, so here is a slide that's kind of helpful just to remember. So the degrees of freedom, remember, like, there's this tendency for people to think the degrees of freedom is just n-1, but it's not true because it changes depending on how many groups you fit means to and things like that, and depends on what test it is. So this is kind of a helpful slide, hopefully, for you guys. So for the one sample t-test, we see that the degrees of freedom are n-1. Okay, we've got one group with one mean. For the repeated measures t-test, it's the same, n-1. For the independent groups t-test, like the one we just did, the degrees of freedom is n1 plus n2 minus 2, or alternatively, it's n1 minus 1 plus n2 minus 1, which is, like in the previous example, that was, there are 10 individuals, so 10 minus 1, 9, plus 10 minus 1, 9. So 9 plus 9, the degrees of freedom are 18. All right. So that's as much as I'm going to say on the t-test. We're using it routinely. Most commonly, we would use it to compare the means of 2 groups. Okay, either an independent design, like the one I've just gone through, or the repeated measures design. Let's move on to ANOVA. So we use t-test to compare means of 2 groups. What if we want to compare more than 2 groups? We can use ANOVA for that, okay, and that stands for analysis of variance. So you can use ANOVA to compare 2 means. It will work like a t-test, it works fine. But routinely, people just use a t-test if you've got 2 groups, and ANOVA for if you've got 3 or more. Okay, so this is the second inferential statistical test we're going to talk about for comparing groups. And it's actually a group of statistical tests that look at differences between group means in a sample to make inferences about differences between population means, between group means and the population, sorry. And it actually, rather than directly comparing means, it compares variability about group means. Okay. So I guess a question that this raises is if we have more than 2 groups to compare, why don't we just use multiple t-tests? Why don't we compare, if we've got 3 groups, why don't we use a t-test to compare groups 1 and 2, t-test for 2 and 3, t-test for groups 1 and 3. The problem with that is we're doing multiple comparisons, and ANOVA, the useful thing about ANOVA is it controls for type 1 error when we're making multiple comparisons. So hopefully this finding a place to leave this in was tricky, but I need to say it somewhere, so I thought here's probably about as good a place. So there are potentially two different types of error or mistake we can make when using p-values to assess statistical significance. So remember, our threshold for significance is p is less than or equal to 0.05. Whilst this is low, it's not zero, and we will occasionally make mistakes. Okay, we could make a type 1 error where we reject the null hypothesis when it's actually true, and this should happen about 5% of the time with an alpha level of less than 0.05. We can also make a type 2 error where we don't detect a significant difference, even though there is one. In this case, our study is underpowered, so we're making a type 2 error. We're failing to find differences when they actually exist. And when we make lots of comparisons to look for differences between more than two groups, or more than two group means, by repeatedly doing t-tests, for example, we increase the chance of making a type 1 error. Okay, so multiple comparisons and the family-wise error rate. Hopefully this is stuff that many of you, it's revision hopefully for many of you, but here's an example to illustrate how this works. The ANOVA test for significant differences between 3 or more groups whilst group means, whilst keeping alpha at p is less than or equal to 0.05. It's called an omnibus test, essentially. And it does that without increasing the type 1 error rate or reducing the power and leading to a type 2 error rate. So it's a real fine balance when you're conducting research as to, and statistical analyses as to whether you're trying to trade off committing a type 1 error with committing a type 2 error, that type of thing. Okay, so if you look at this particular slide, this example, so imagine we have 7 drugs to treat depression or some sort of disorder. It would be possible to conduct 21 t-tests to test the effect of every drug against every other drug. So drug 1 against drug 2, 1 against 3, 1 against 4, 1 against 5, 1 against 6, 2 against 3, 2 against 4, etc., etc. Okay, so that would mean doing 21 different t-tests. And if we set the alpha level at 0.05 by convention for each comparison, then what's known as the family-wise error rate becomes very high. So the family-wise error rate for alpha, the way we'd calculate this probability is 1, which is the total probability, minus 0.95. So the complement of our 0.05 threshold. So there's the sort of 95% level. So 0.05 raised to how many tests we're doing. So the exponent there, 20 in this particular formula, is p equals 1 minus, and then in brackets I've got 0.05 to complement the opposite probability to our alpha criterion, 0.05, raised to the power 21. How many, the 21 tests we're doing to compare these 7 drugs. And if you do the calculation on that, what you get is a family-wise error rate of 0.64. So a 64% chance of making a type 1 error. So a 64% chance of you finding a significant difference, even though there isn't one. Okay. And ANOVA test if there is a significant difference between any of the means with one statistical test whilst keeping that alpha level, stopping this inflation in error. Okay, so ANOVA is another null hypothesis significance test. We set up ANOVA with a null and an alternative significance test. So usually the null hypothesis H0 for ANOVA is that all population means are the same, and any difference between sample means is simply due to sampling error. Okay. The alternative, or H1, is the population means are not all the same, and there's at least one significant difference between the groups. So how does ANOVA work? The basis of ANOVA is the F ratio. Okay, this is a ratio of 2 estimates of population variance, both of which are based on the sample. So the first one is the within group variation, or the within groups variance or error. So within each sample there will be variation. This will be how much the scores will vary around their respective group means. So if we've got 3 groups, we work out the mean of each group, scores will vary around those means. Okay, that's the within groups variation, or often referred to as the error variance, or the mean squared error in the case of ANOVA. I'll say more about that in a sec. Okay, the other estimate is the between groups variation. So if the null hypothesis is false, and then the mean of group 1 isn't equal to the mean of group 2 or group 3, or at least one of them isn't equal to one of the other groups, there will be variation between the group means. Yeah. If there was no difference, those, say we've got 3 group means, they'd all be about the same. But if we get a significant ANOVA result and the null hypothesis isn't true, there'll be a difference between group means. And what this quantifies the between groups variation is how much the group means vary around the overall mean, or the grand mean, for the whole sample. You can use ANOVA to test. You can use it for independent groups designs, repeat measures designs, or even mixed research designs where you've got variables that are repeat measures and some that are independent groups. Okay, in order to use ANOVA, I'll come back to the F ratio in a second, but in order to do an ANOVA test, it's a parametric test. So there are a couple of assumptions, key assumptions we have to meet. The first is that of normal distribution. So scores for the population and within groups have to be normally distributed. And the second is something called homogeneity of variance. For repeat measures designs, it's called sphericity. They're a similar sort of thing. And then what this means is that the groups come from populations with the same sort of variance. So what it means is if we've got 2 groups, the variance around the group mean for group 1 is kind of similar to the amount of variability around the group for the second or the third group. There's not a huge difference. You've not got this much variability for one group, this much for another group. Okay, and we need to meet these assumptions in order to get valid and accurate results out of an ANOVA test. So I'll say more about homogeneity of variance and the tests we can do for it to see if we can meet that assumption. I'll say more about them in lab classes. But if we meet these assumptions, these 2 assumptions, it's usually safe for us to use the ANOVA parametric statistical test. All right, so the F ratio, as I said, this is the basis of our ANOVA, and we get a statistic from an ANOVA called an F statistic, and it's calculated by basically taking the between groups variability and dividing it by the within groups variability. So it's a ratio between groups to within groups error, essentially. And the F value will be 1 or less if the null hypothesis is true, if there is no difference. If there's no difference between the means of the groups, then the means will be quite similar, and the result of this kind of ratio, this division calculation that they're carrying out here, will be 1 or less. If it's 1, then the amount of variability between the groups is exactly the same as the amount of variability within the groups. Okay, so they've all got the same mean and they've all got the same amount of spread. If the value of F is greater than 1, then it's likely that the null hypothesis is true and the means differ. And if that's the case, if the difference between groups gets larger than the variability within the groups, then F will be greater than 1. Okay, so if how much the means differ is much greater than how much variability there is around the means, so the groups and their variability don't overlap, then we'll have an F value that's much greater than 1, and it's likely to be statistically significantly different. So the F statistic has a p-value associated with it, and it's based on the degrees of freedom. that we have for the between groups and within groups variance. Okay, so we can look up those p-values when if we work out an F statistic, do an ANOVA test, we can work out what those p-values are by looking them up in tables. There are critical values for what's known as the F distribution, depending on our degrees of freedom. We don't need to do that, so we've got software to do that for us. SPSS will do that. I don't want to get too technical here with regards to the formulas for ANOVA. In the lab classes, I will hopefully make this all clearer by drawing out some of these things. So using scatter plots, and hopefully that will clarify things for people. See how this works and what it's actually doing. So just to help with that though, here is to illustrate this. Here is to illustrate really how straightforward it is to work out what and how, like get an ANOVA result using software. This is the ANOVA summary table that you get in from SPSS output. Okay, so the mean square there, that's the column that's labeled mean square, that is the variance, and it's for some reason it's called mean square in ANOVA. So you've got the mean square between groups, mean square within groups, or mean square factor, mean square error. You'll sometimes see them referred to as. Not sure why for ANOVA the variance has been given the name of mean square, but that's what it is. Okay, so if you remember from the variance calculations last week, when you work out the variance, you get this single, you sum the squared deviations from the mean, and that gives you, and then you add them all together, and that gives you a single value called the sum of squares. So the sum of squares column there shows what the sum of squares value for the between groups variability and the within groups variability for this particular test. Let's say this was a test, I don't know, comparing three means, I think, more than likely. If you divide the sum of squares by its respective degrees of freedom, then you'll get the mean square. Okay, so if you look at that first column between groups, if you take 794,408, 794,408.154, if you take that and divide it by 3, which is its respective degrees of freedom in the next column, what you will find is you get the mean square, 264802. So 264,802.718 is what you'll get. And then if you take the within groups sum of squares, 7613921.23, divide that by its respective degrees of freedom, which in this case is 456, you will find you get the mean square in the next column, which is 16697.196. Okay, if you then take those two mean squares, so the between groups and the within groups, and you divide the between groups by the within groups, that's the ratio of between groups to within groups, that gives you the F value. So if you took 264802.718 and divided by 16697.196, you will find you get a value of about 15.859, which is the F statistic. With such knowledge, you could then look up, use an F distribution table, look at the critical values and figure out what the probability is. But luckily, in this case, SPSS gives us the statistical significance in that column labeled SIG for significance, and it tells us that it is less than 0.001. So a highly statistically significant difference. Right. And at the bottom of this slide here, you can see how to form the report on F statistic, and you've got, so like the T statistic, you've got F showing what the statistic is, and then in brackets you've got the degrees of freedom. Notice this time you've got 2 degrees of freedom, one for the between groups, yeah, which is 3, and one for the within groups, which is 456. So you've got in brackets, so you've got 3,456. And then equals 15.86, so they've rounded the F value to 2 decimal places, then a comma, then P is less than 0.001. Right, so that's how you would form the report. We're going to go a little bit beyond that, I think, and we'll probably look at effect sizes for that. add that onto the end when we formally report these things. But I shall talk about that shortly. Right. Quickly, I'm going to have to rush through these because we're running out of time. So post hoc tests. So that was all very good, what we saw on that slide. We saw whatever the three groups, maybe there were three groups being compared there, and what we found was there was a significant F result. F equals 15.89, P is less than 0.0. So we've got a significant ANOVA test. That's nice, but a significant ANOVA result only tells you that at least two group means are significantly different. It doesn't tell you about the difference. So if you've got three groups, the only thing a significant ANOVA result tells you is that the largest mean and the smallest mean are significantly different. It doesn't tell you if the ones in the middle are different to the largest, or the ones that are in the middle are different to the smallest, that type of thing. Okay. So we need some way to test, to assess where the significant differences are, which groups are significantly different to which in our three or more groups. And to do that, you do post hoc tests. And this means multiple comparisons and the possibility of inflating type 1 error. Okay. Post hoc tests, thankfully, do these comparisons for us whilst trying to strike a balance between type 1 and type 2 error. So they do it by adjusting the alpha level for a significant result. So we're not committing a type 1 or a type 2 error. There are lots of different post hoc tests. There are about 18 you can choose from when you use SPSS to analyze results. And they vary in how strict they are, how strictly they control, how strictly they control for a type 1 error at the expense of maybe a type 2 error, or vice versa. Okay, so if you use a post hoc test that is too strict, then although the chances of making a type 1 error are small, the chances of making a type 2 error increase. Likewise, if you use a post hoc test that's too lenient, then although the chances of making a type 2 error are small, the chances of making a type 1 error increase. There's no one answer to this, and you as the researcher need to use your judgment when choosing a test. Okay, so there are lots of different tests, and there are situations in which you might use some rather than others. So what I would suggest is when you're learning about post hoc tests, you look at the Andy Field textbook in its section 12.5 in the 5th edition, which is the most recent edition. Okay, I'm just going to mention a couple here that are kind of useful and commonly used. So probably the most commonly used is the Bonferroni correction, or the Bonferroni t-test, sometimes referred to as Dunn's test. Rarely hear it called that now. But one solution to this issue of making multiple comparisons by doing multiple t-tests is to change the level of alpha to make the test more strict. And the Bonferroni procedure or correction is just you take your alpha criterion level and you divide it by the number of comparisons you're making. The effect this has on the family-wise error rate is to keep it strictly to the 0.05 level. Okay, so so our threshold for significance is 0.05, and if we were doing three comparisons, all we do here is we divide 0.05 by 3. That will give us a figure of about 0.0167, and we take that value, 0.167, as our new threshold for significance. So if we then compare two of the means from our significant ANOVA, and the comparison shows a p-value less than 0.0167, we'd say that was a significant difference. But if it was more, we'd reject it. Okay. So yeah, essentially, you adjust your significance level when you're doing your multiple post hoc tests, and then you conduct whatever your t-tests are and evaluate them against this new level of alpha. Okay. So with regards to the Bonferroni t-test, it controls for type 1 error very well, but at a cost of type 2 error. So it's quite a strict test. And apparently it has more power when you're only using a small number of comparisons. So if you're only doing 3 comparisons. Some other common post-hoc tests here are listed on this slide, and they go down from the most strict to kind of the least strict. There are loads more other than this, but the Bonferroni t-test, that's the one I've just demonstrated, that's quite a strict test. Slightly less strict that is the Tukey's honestly significant difference, or Tukey's HSD. Slightly less strict than that, more lenient, is Newman-Kuhl's, often referred to as SNK or student Newman-Kuhl's. And then at the more lenient end, you've got something called Fisher's least significant difference. Okay, so Fisher's LSD is the most lenient. It is basically just doing 3 t-tests without any controls. And it's been suggested that you can safely use it when you're comparing a small number of groups, like 3 groups. I would say don't use Fisher's LSD, because even with 3 groups, it inflates the type 1 error rate to about 14%. Okay. Tukey's HSD apparently might be a good alternative to the Bonferroni test. It controls well for type 1 error, and it's kind of better than Bonferroni if you've got lots and lots of comparisons to make about it. Okay. We'll cover more on these post-hoc tests in the lab classes. And there are, in addition to sort of some of these more common post-hoc tests, there are post-hoc tests that you would probably use as well if your data doesn't meet the assumptions of homogeneity of variance. So there are more robust ANOVA and post-hoc tests, and we'll cover those as well. Right. So just to finish off, a couple of things. I'm over time, so I'm going to have to speed this up when I give the actual lecture. So today we've kind of looked at models for inferential statistical tests to compare groups, or compare group means essentially. And those models look a little bit like that, sort of that formula I've put on the top of the slide there. So we've kind of divided some sort of variance that's explained by the model by the amount of error or variance not explained by the model. That's certainly the case with the F ratio. So that's a measure of the model effect divided by the error. And that's given us some sort of test statistic, be it T or F. What we've then done is, with our test statistic, we've compared our test statistic against critical values for normally shaped distributions to get a p-value. So we could be using, for F, it could be, we're looking it up in critical F value tables. For the T distribution, we're looking at values, p-values associated with the T distribution. And we're using those to tell us whether the test statistic we're getting is statistically significant. It's showing a statistically significant difference depending on our group's degrees of freedom. This tells us whether or not our groups were significantly different. It's basically saying if we find a statistically significant difference, then we have a difference between the groupings. Yeah, but it doesn't tell us if that difference is interesting or meaningful. Okay, so to answer that question, we need measures of effect size. Okay, so effect size. So statistical significance doesn't tell us anything about the importance of an effect. Measures of effect size tell us about the practical significance of our results. So effect size is a kind of measures of effect size are standardized ways of measuring the magnitude or size of the observed effect that we've seen with our testing. Okay, standardized means that we can compare effect sizes across different studies that might have used different scales of measurements. So there are various different effect size measures. One you've probably all encountered before is the correlation coefficient. It's a measure of effect, essentially. Partial eta squared is a commonly used effect size with ANOVA, and SPSS will calculate this for us. And it's useful because rough benchmarks exist that allow us to assess whether the effect, our result shows an effect that's small, medium, or large. Yeah, and partial eta squared is essentially a measure of the variance explained by our model. Okay, so run out of time, so I will leave it there. I've gone about 10 minutes over. Okay. References, I've just put one reference on this slide at the moment. I might dig out some more if I can find some that I like. But if in the most recent version of the Andyfield textbook, 5th edition, chapters 10 and 12. 10 covers comparing two means, so t-tests. 12 covers ANOVA. Those are good. Those are the resources that are kind of most useful if you want to do some reading and learn more about these tests where we're comparing means. All right, we'll go into more depth this stuff in lab classes. So I shall see you soon.